Oxidative phosphorylations. Sarah?
See, yeah,
No? Zach?
See, I thought oxidative phosphorylation was like a last step of glycolysis.
Last step of glycolysis?
My bad. Of respiration.
Ah, very good. There are none. Trick question. Perfect. Perfect. So there are no oxidative phosphorylations. That occurs as a result of electron transport, right? The name itself, oxidative phosphorylation, refers to the fact that we have redox reactions, reductions of the NADH, FADH2, which is ultimately going to drive the formation of ATP and the electron transport chain. So there are none. Substrate level phosphorylations, just one, right? This one right here. So succinyl-CoA to succinate, the succinyl-CoA synthetase drives the formation of GTP or ATP in some species. Okay? So just one substrate level phosphorylation. Recall though that how many turns of the Krebs citric acid cycle, TCA cycle, do we get per glucose?
34.
2. Oh, 2. Right? Because how many pyruvate get produced at the end of glycolysis? 2. And there's going to be 2 of these turns here. Just to kind of, for those of you keeping track, no. But if you wanted to, that's why I've put up this lovely table here. Yeah, you know, I also knew what women wanted at one point in my life, and I've completely forgotten, so hence why I've gotten multiple ultimatums over the past few years. So, the information is important, right? All right. So, glycolysis, how many ATP? All right, 2. NADH produced?
2.
FADH? 0. Intermediate step, this is the one from pyruvate to acetyl-CoA. Big thing on this one is the NADH that could produce. Some people lump this in with Krebs cycle. I don't really care which way you do it, which is in part why I'm not really worried about you knowing the numbers here. But Krebs, 2 ATP, 6 NADH, 2 FADH2 per molecule of glucose. So total numbers are this. If we could describe, maybe in one term, what the ultimate, 10, not P.
Or 4. Instead of 2. 2 plus 2 is...
Thank you. I also have lost the ability to add. All right. If we could describe, maybe in a single phrase or word, what the major theme of Krebs cycle is, what is it? She did. I heard it too. I think it was the right answer. It's redox reactions. That's the major theme. Because if you take a look at the majority of what gets produced in this, we've got 8 coenzymes being reduced in this process here. So the ultimate goal of Krebs cycle is to take every single carbon from that molecule of glucose and completely oxidize it. That's where we get the CO2 in respiration from, is that oxidation of those carbon molecules. So those reduced coenzymes then, what happens? Where do they go? What do they do? In other words, what happens to NADH after this step? Electron transport chain. Where does that occur? Okay. Okay. Nobody said mitochondria, right? No, because some people did on their responses, and I had to make sure that we get that out of the system. All right. I usually like to draw analogies here. So think of mitochondria real quick. One time we'll talk about eukaryotes today. So mitochondria have that outer membrane. You'll recall they have these intermembrane folds, matrices in the middle, intermembrane spaces in between. Gram-negative bacteria, 2-membrane system as well. in Krebs cycle is going to occur in the matrix. In bacteria, occurs in the cytoplasm. Essentially the same location, right? And we just said that in gram-negative bacteria, or bacteria in general, sorry, this inner membrane is the location of that electron transport chain. Same thing with mitochondria. That inner membrane there is location of the electron transport chain. Where do the electrons get pumped?
That's me. I was gonna say that ain't my reason. I got that one.
Real quick story since we're listening to music right now. When I was 10, so I played Little League Baseball. Remember I was pitching one day, and there was a jazz band at a band shelter playing. There was a whole concert going on. And as I'm setting up to pitch, I actually started like beating to the music. The umpire started laughing so hard, and I was pissed off because I wanted to know if I had thrown a ball or a strike. So, and all the fans were laughing, and I was like, "What is it? Ball and strike?" Anyway, I'm competitive. So, Sarah?
I just have a quick question about oxidative phosphorylation.
Yes.
For the redox reactions, when you're looking at the Krebs cycle, you can tell what the reaction enzymes, it's like when the
Yes. So there are none in Krebs cycle, right? Really, when you're thinking oxidative phosphorylation, what you're looking for is electron transport chain and ultimately the ATP synthase. Because that's the only place where this oxidative phosphorylation is occurring. If you're asking about substrate level phosphorylation, what you're looking for is for ATP to be made or broken down and for some other substrate to be involved in the process as well. So let me go back real quick. I'll redraw these. So like in glycolysis, for example, glycolysis has a really couple of great examples of substrate level phosphorylation. So here in this picture, we've got phosphoenol pyruvate. This phosphate right here gets transferred onto ADP to form ATP, and it produces another new product, pyruvate. So really what we're looking for is just transfer of phosphate. In oxidative phosphorylation, on the other hand, the phosphate comes not from another substrate. The phosphate's actually just kind of floating around in solution. So let's skip ahead real quick. In this case here, what this is showing is that we have 3 ADP plus what's labeled as PI, inorganic phosphate. That phosphate's just kind of floating around in the cytoplasm when this is taking place. So it's not transferred between a substrate and another substrate. It's just that phosphate's coming from the atmosphere in this case. Yes, exactly. Okay. Redraw these real quick. Okay, so we said that the electron transport chain is here. Where do hydrogen ions accumulate in the mitochondria? So as the electrons are moving through the electron transport chain, where do those hydrogen ions get pumped? What's that? Yeah, it's this intermembrane space. It's this region between those two membranes. By analogy, the location is the same place in a bacterial cell. So between this outer and inner membrane on a gram-negative or just outside that inner membrane on a gram-positive, we'll find those hydrogen ions accumulating. Okay. So keep in mind as we now just zoom in on part of this, and we're just going to focus on the chain here. Okay. So high energy electrons from NADH and FADH2 will go into this chain. Cross that out real quick. Hope nobody saw it. NADH drops off the electrons, becomes NAD. What kind of reaction is this? Redox reaction. So NAD is being oxidized, loss of electrons. This complex 1 here is being reduced. What kind of enzyme is this complex? Don't think too hard. Kinase dehydrogenase isomerase. It's a dehydrogenase. The other name for this is, in fact, NADH dehydrogenase. Okay? So, we have a dehydrogenase, and you can kind of think of it as each of these individual pieces are involved in redox reactions. So as those electrons move through from one to the next, let's just follow electrons here in this first case. NADH drops them off into this NADH dehydrogenase. This gets reduced. Hands off the electrons to ubiquinone. This becomes reduced. This is now oxidized. And we just have a series of those steps. Electrons from ubiquinone move to cytochrome B and cytochrome C1. This becomes oxidized, these become reduced, and so on and so forth. Okay? So it's just a set of redox reactions the whole way through. One of the things that takes place through here is that as the electrons are essentially being pulled, each of these complexes, ubiquinone is more electronegative than complex 1. This cytochrome B, which is complex 2, is more electronegative than ubiquinone, and so on and so forth. Anybody remember what electronegative means, or the principle of electronegativity? It's a great review question for the exam. So what is electronegativity? Contributes to polarity. Okay, so if something is more electronegative, let's say we'll take hydrogen and oxygen, right? Oxygen's more electronegative than hydrogen. In that covalent bond, where are the electrons? Closer to the oxygen, because it's more electronegative. The pulling effect here of those electrons is due to the electronegativity differences in these complexes. So essentially, since ubiquinone is more electronegative, it's going to pull onto those electrons in this direction. Cytochrome B1 is more electronegative than ubiquinone. It pulls those electrons. And we keep going. What's the most electronegative molecule in this whole thing here? I heard somebody say it. Oxygen is it. Oxygen is the final electron acceptor. Therefore, to be able to pull those electrons out of this electron transport chain, it has to be the most electronegative of all of these complexes and molecules in this process here. As those electrons are moving from one complex to a next, they also lose energy. That energy is used to do what? Pump what back into the cell? Pump them, and not pump them in, but actually pump them out. So it's pumping them from the cytoplasm to the periplasm in a gram-negative, just outside the cell.
So you get the hydrogen out of the cell, it's an active transfer.
Yes.
And then you get it in, it's only passive.
Exactly, exactly. And so you'll remember some of the things that we talked about active transport. Typically, molecules are moving with or against their gradient. It's against. So these hydrogens are going from areas of lower concentration to an area of higher hydrogen ion concentration. That allows for then the passive diffusion to be with the with the gradient. Go ahead, Seth.
And then when all of those, what are those called? The blue things? Yeah. Complexes? Is that what you call them?
Yeah, yeah.
When all those are oxidated, what does that do? Or what does that do?
So so when all of them are oxidized, what does that do? It allows them to be able to accept the electrons, so that the train. I mean, keep that in mind. We'll come back to that. Actually, it can be important. It's actually a control of microbial growth. All right. typically the rule is NADH produces about 2.5 ATP for every NADH that's available, 1.5 ATP for every FAD that's available or oxidized. Obviously, in this case, oxygen is the final electron acceptor, and then as those hydrogens are moving through, the ATP synthase drives a turbine that allows for the ADP and the inorganic phosphate to basically bond together covalently. So that's our creation. One thing to point out here, I should go back to, I'm going to draw this real quick again. One of the things that's really nice with bacterial cells is that, no, damn you. All right. So mitochondria, bacterial cell. Mitochondria, where does glycolysis occur in a eukaryotic cell? It's actually in the cytoplasm. It's not even in the mitochondria. And so what that means is in order for Krebs cycle to occur, pyruvate actually has to be shuttled across both of these membranes into the matrix. That process requires energy, requires ATP to occur. Because bacteria don't have to do that, because Krebs cycle and glycolysis occur in the same place, bacterial cells actually get a couple of more ATP per glucose molecule than do eukaryotic cells, because they don't have to do that shuttling process. Just wanted to point that out real quick. All right. Questions on electron transport chain. And what we've looked at so far is aerobic respiration, where oxygen is the final electron acceptor. How does this change when we talk about anaerobic respiration? So what's different in anaerobic respiration? That's the biggest difference. Oxygen's not the final electron acceptor. What is? Sulfate itself, yeah, would be 1. Nitrate would be 2. Carbonate's the third one. And I'm going to just skip to this table real quick. These are actually in order of which one gives the most energy per molecule of glucose, which one actually gives the least amount. So what I'm trying to bring up here is actually between aerobic and anaerobic respiration, in terms of the amount of energy that's harvested in the form of ATP, aerobic respiration does the best because oxygen is the most electronegative. That allows for the greatest release of energy from those electrons from NADH and FADH2 over time. Nitrate's the second best. It's the second most electronegative of the acceptors, which allows for it to be pretty, which allows for a pretty good yield of energy. Carbonate out of them is the worst one. And now I'm angry. Okay. I'll figure this out eventually. So nitrate again is the best of these anaerobic acceptors. Sulfate, second best. And then carbonate is the least of them. Okay. For these, the electron transport chain actually changes a little bit. There are different complexes for the process of electron transport for where nitrate is the final electron acceptor or where sulfate is the final electron acceptor. I don't want to bore you or to make you think that you need to know all of this information as much, but I do want to point out, in the case of nitrate, nitrates, the final complex in nitrate is known as nitrate reductase. It actually bypasses the use of these two complexes and replaces this one here. So nitrate reductase will accept electrons directly from ubiquinone, reduce nitrate to nitrite, and because there are fewer complexes, because that allows for fewer hydrogen ions to be moved across the membrane, And that's what leads to the lower amounts of ATP that's yielded in the end. Okay? So I just wanted to point out why this occurs. Let me show you a quick video. Let's see if I can pull it up. Okay, so I went to Kohler Andre like 3 days after we had our first day, or our first week of the semester. I'm going to show you a video of what I saw. This is not my video. I will not take credit for it, but this is like what I saw. So that's sand. Okay, so there are the waves. Do it one more time. What colors of sand are there? This is not a trick question. It does have microbiological purpose. What?
Like in
Yeah, just like in this picture right here.
black and tan.
Black and tan. Why?
That's just the way Mother Nature created that sand.
It actually is the way that Mother Nature created it, but Mother Nature didn't create the sand to be that way. What test have we done in the past couple of weeks?
Sulfur.
Sim test. The bacteria that you'll find on Lake Michigan and some of these other areas are sulfur reducers, or sulfate reducers. The sulfovibrio is one of them. This black sand is actually a reaction between the iron in the sand and the hydrogen sulfide that gets produced as a result of that anaerobic respiration. So if you were actually to dig down deeper into the sand, you would find a higher concentration over time of the black color that you see. What's true about that environment? If you dig down in the sand, oxygen there, oxygen not there? It's not there. So the options for the organism in that case is they can't perform aerobic respiration. They have anaerobic respiration as an option or fermentation, which we'll talk about on Friday. But that color you observe there is really just from that sulfate. And if you were to take a big whiff of it, just pick some up, smell it, it smells like sulfur, rotten eggs. Okay, so that's where that smell and where that color comes from. Just wanted to bring that up. Sulfur reduction. Okay. The sim test tests for... Go ahead. So what does the sim test do? And some of you will see it later today. So it's sulfur, indole, and motility. The sulfur part is the hydrogen sulfide that gets produced by some of the bacteria. It will react with the iron to produce iron sulfide, and that's where that black precipitate color comes from. Okay, so it's an indicator that that microorganism can actually perform anaerobic respiration, and not only that, but that in its form of anaerobic respiration, it's using sulfate as a final electron acceptor. I want to point out that for these three modes of anaerobic respiration, with nitrate, carbonate, or sulfate, organisms typically have, if they can do anaerobic respiration, they typically are going to use one of those final electron acceptors. It's rare that all three can be used as final electron acceptors. And again, the determination of what allows for one or the other to be used is dependent on the proteins that are within this electron transport chain, or the genes that encode for those proteins by the microbes. Last point I want to make right now is when we're talking about respiration, respiration really is referring to Krebs cycle and this electron transport chain. In opposition, we have fermentation, which is just going to be glycolysis and then a reduction of pyruvate, oxidation of NAD. And that's what we'll talk about on Friday. Any questions? let's do some practice. All right. I'm going to actually use, for this practice, an old test question. Yay! Oh, I don't want to go there. So this is one that's focused on what happens if the electron transport chain gets screwed up, basically. So rotenone binds to complex 1, NADH dehydrogenase, in the electron transport chain and prevents it from passing electrons to ubiquinone, which is the second complex. One might consider rotenone to be a non-competitive inhibitor. Describe the structure of such an inhibitor relative to the substrate and where it binds an enzyme. So that was part 1. Part 2 is explain what effect treatment with rotenone will have on respiration. And you should be including terms like electron transport chain, hydrogen ions, production of ATP. Okay? Go ahead. Have fun with it. Yeah.
Inhibitor can bind to the enzyme with or without a substrate
Okay.
at different places on the enzyme, like at the same time.
Okay.
So it just changes the shape of the enzyme, I guess,
Mm-hmm.
and the active site, so it runs less effectively.
Good. That's great. Where does it bind? What's that? Yeah, allosteric site. That's like the one thing that I would add to it, but otherwise that's a great answer. Okay? You probably would have gotten full credit for it anyway. Because it hits all the major points, all right? What effect does it have on respiration? I know I heard a correct answer up front. Zach, go for it. You said you had it, so go for it.
Oh yeah,
You didn't say anything.
so we said that since the enzyme won't, since it's that's involved in redox reactions, the hydrogenase, the enzyme's not going to correctly be able to pump or not pump, but pass electrons.
Okay,
or
good.
get electrons.
Good.
Because, the shape won't match up. So.
Perfect.
Yeah.
So if it's unable to, let's go back to that. So if it's not able to do that, does a hydrogen ion gradient get created?
No, because there's no NADH.
Good. And then there's no ATP in the end either, right? Perfect. So in this one, what I would be looking for is for you to kind of connect all of these ideas. I'd be looking for somebody to say, to walk through this logically, basically, and say, damn voice thread. That's what I would expect.
Your answer.
I thought
Just your guess.
I thought voice thread was the best.
when it is, but you can hate best friends from time to time. All right, so what I'd be expecting in an answer would be to say, just as you started out, it's not going to be able to accept those electrons from NADH. Therefore, the electron transport chain will not have redox reactions. Knowing that those electrons are involved in pumping the hydrogen ions, you would go along and say, no hydrogen ion gradient is created. And because that's not occurring, no ATP is being created. What would the cell do in response? That quite possibly, it depends on what kind of organism it is, right? If it's what we classify as an obligate aerobe, one that requires oxygen to live, it dies. If we get treated with rotenone, we die, okay, if that's strong enough concentrations. You basically die from asphyxiation. A&P term. Who said we didn't talk about A&P in here? Me. 2, if it's an organism that's facultative, and what we mean by that is it can do both aerobic and anaerobic, then it might switch metabolism. One of the ways that it might switch would be towards a more fermentative process where it still can produce ATP and still get oxidation of the NAD molecules as well. What's going to happen at Krebs cycle? Would Krebs continue? No, why not? You can't go any further, yes, but what do we actually mean by that? Think about, so what reaction is being stopped here again? Rotenone blocks oxidation of NADH, right? And it blocks its oxidation to NAD. What do we actually need to drive Krebs cycle? We need the oxidized form of this coenzyme here. So if there's no NAD around, if it's all in its reduced form, these reactions can't take place, and therefore Krebs cycle shuts down. That's why we say that Krebs and electron transport are mechanistically linked with one another. Okay. Shall we try one more quickly? All right. Hops actually have antimicrobial activity. Hops have what we call isohumulones, and isohumulones are also known as ionophores. Neither of those terms you actually need to know what it means, but you need to understand, or what I would provide you is the information of what's the function of that ionophore. Basically, it takes hydrogen ions and it allows them to passively diffuse through the membrane. What's this going to do to energy yield? What's it going to do to the electron transport chain, hydrogen ion gradient, yada, yada, yada. Okay? Take like 2 minutes to try and answer this one.
If it's inactive, like if it's not present or what?
So if the isohumulones were present, what would that Right, so they go out through the first and the third and the fourth, right? And they also extend passively through. So if they're coming in passively, something happens. Does that increase or decrease the amount of hydrogen coming?
So it's just saying what's there, it's
Yeah, that's a great.
You can miss me with that.
Will that affect the electron transport chain?
no, it is not or like bringing them out.
No, oh yeah, bringing them out or just the electrons themselves flowing through.
That's a concept. Oh, that's when it's like.
All right. What's going to happen to the yield?
No ATP.
No ATP? Why not?
Because the H ions won't go through ATP synthase to.
Perfect. Or at least less frequently. So there's less ATP that gets produced. Perfect. What about the electron transport chain itself? So we saw with rotenone it would be stopped. Is that going to be true here as well?
Wouldn't it just be slow, just not stopped?
Good question. I don't know. Actually, I do know, but. So, not the citric acid cycle. Does the electron transport chain continue? If isohumulones are affecting that hydrogen ion gradient. Here's the way to think of it. What did rotenone actually do? It blocked the flow of electrons, right? It blocked that first redox reaction from taking place. Does an isohumulone do that? No, actually. And this is kind of the thought process here that maybe I would want you to think about is, it's not hydrogen ions that's controlling the flow of electrons. It's actually the electron flow that's controlling the hydrogen ion gradient. And so if we block that hydrogen ion gradient from getting created, those electrons still flow. NADH still gets oxidized. Krebs cycle still occurs. Cells that are exposed to isohumulones basically starve to death. They continue to consume, but they aren't able to produce any ATP from it, or at least enough to sustain life. Okay, so that's typically what happens. So it would lower the amount of ATP that gets produced because overall, because the hydrogen ions can now pass through more locations in the membrane, that limits how much will flow through the ATP synthase, but the flow of electrons remains unchanged. Okay. All right, so let's just summarize real quick major themes of the day. What are the major couple of points that we talked about? Oxidative phosphorylation, electron transport chain, H+, all of that kind of combined. Think of that as a single set of information, okay? And really what's important here is being able to make the connections between the ATP, the hydrogen ion concentration, and the electron transport itself. Anything else? If anything, just what's the major role of the Krebs? Reduction of those coenzymes. Okay. Homework for Friday is a group wiki. It's comparing aerobic, anaerobic respiration and fermentation. I think it's a great kind of cap of today, but also a really good look ahead. If you're in a group of 3, each group member take a different column. If you're in a group of 4, each member take a different row of it. Okay? I think that actually will work out. On Friday, our major goals Friday are to talk about fermentation, talk about phenol red tests, which some of you will actually, you all would be setting up today. And we'll talk about the interpretation of that and how it tells us about, or what it tells us about fermentation. And then I would also like to go over the format of the first exam so that you are aware of what it looks like, okay? Not what the questions are, they are not written yet, but what the format is. No, I wouldn't be
Is Monday like a regular day?
Monday we're still going to talk to,
Like close some ideas.
we're actually going to talk about something. So it's going to be, it'll be a little bit of new information, but largely we're really just going to kind of be working with these set of themes that we're doing. So I wouldn't necessarily call it a review day, but in some ways it kind of is.
Okay, a while. Yeah, but I was looking at the calendar, so I was like stressing with that.